Hello students, I hope you're having a great day. Today we're going to go over flower dissection. This is our first dissection in biology. You should have this worksheet and some colored pencils with you, something to write with as well, a pen or a pencil. And as well as this, you will have a dissection tray with your flower and these tools available to you. So make sure you have all of that before we begin. Okay, so the first thing we always do in lab is we read our instructions. So we will start here at the top and begin. It says flowers have both male and female reproductive structures. In this activity, you will take the flower apart to identify those structures and examine them closely, as close as if you were a bee coming in for some nectar. Here is a list of our materials, and once you have those, then we can begin the pre-lab, which is a concept map. So in our concept map, we have this word bank to use within this diagram. So the first thing we want to talk about is what is our overarching word that we're using, and that would be flowers for this lab. Notice that flowers have both female and male structures. Some flowers have just female, some flowers have just male, and some flowers have both. So we need to know all about the structures that are in flowers. So let's look at female structures. The first female structure, or all the female structures together, are called carpels. And the carpel is made up of three different parts. First is the stigma, then the style, and last is the ovary. Inside the ovary, you have something called ovules. Another name for ovules are eggs. Okay, in the male structure, the whole male structure is called a stamen. And a good way to remember that this is a male part is stamen has the word men in it, meaning, of course, male. Two parts to the stamen are the anther and the filament. Again, the word men in filament. The anther produces something called pollen, and inside pollen you have sperm. Okay? So that is the parts of the flower. Flipping over to the second page, notice you have your diagram here of the anatomy of a flower. Notice the stamen is the anther and the filament together. That is the stamen. You have one here and one here. Notice they're in very close proximity to this structure, which is the pistil or the carpel. It goes by both names, which is the female part of the flower. Notice the stigma is this top portion of the carpel. The stalk, or the tunnel, if you will, that goes from the stigma to the ovary is the style. And then the ovary is the enlarged part down here at the bottom, which has the ovules or the eggs in it. So get a flower from your teacher and use one you brought to class, and familiarize yourself with the parts of your specimen using the diagram above. Note that different flowers have different numbers of petals, stigmas, and leaves. So if you look at our example, you notice we have petals and sepals, and we have both male parts, which are the stamen, and we have one female part right here in the middle, the carpel, and these are all of our petals. Okay. Moving on to number 2. To begin your flower dissection, gently pull off the petals and set them aside. Note how many petals are on your flower. Okay, so first of all, I want you to watch what I do, and then you can pause this video and do it yourself. So you're going to take your tweezers and you're going to find an outside petal, and you're going to slip your tweezers all the way to the bottom and firmly just pull off. Okay? Let's see, here's one. Notice it's on the outside, just squeeze and pull. They come off very easily. If other parts come off too, that's okay. Just grab those, lay them down. Sometimes that happens. Not always, but sometimes. And so we have 1, 2, 3, 4, 5, 6 petals. So you want to write that down here, 6. And then the next question says, locate a stamen. The stamen consists of a stalk called a filament and a pollen-producing anther. Carefully remove the stamen. And we have already started doing that because they come off very easily. Just same way, reach and pull. Now, unfortunately, my carpel fell out as well. You should have this structure that is left, just like that. But mine fell out. That's okay. Okay. Using your colored pencils then, draw one stamen in the space provided, label the filament and the anther. So you're just going to come in. Doesn't have to be anything perfect. We're just going to draw in something along those lines. Look at it closely. You'll probably see some pollen, maybe not. Just take a look at it very, very closely. Draw what you see and color it in. Okay? Trying to shade mine just a little bit because it gets a little lighter as it goes down. So then this would be the anther, and this would be the filament. Okay? All right. Moving to the next section. It says use a magnifying glass or stereo microscope to examine the anther and locate the pollen grains. Draw your findings. Okay, so if you look back here really, really closely, I'm going to move my camera down just a bit so that we can come in really closely to those anthers. You'll notice in there there's some yellow stuff, and it might even get on your pad as well. That is the pollen that we're looking for. Take a close look at that, and then on the microscope there's also another stamen. You won't be able to see it in color, but you might be able to see the shape of the pollen. So that's what you're looking for. You're trying to look at the pollen itself and look at how it is located on the stamen. So once you get that back into focus, we'll come back over here. Again, just drawing an anther. And they're just kind of particles. On the anther. Sometimes they stick off a little bit. The whole point is this is what pollen looks like. Just going to color in our anther. Remember what's inside the pollen? Well, what's inside the pollen is sperm. And to me, the anther has always kind of looked like a sperm. If you've ever seen a picture of a sperm, the anther reminds me of a picture of a sperm. So that makes it a little bit easier to remember that this is the male part. Okay, so you can see that well. All right, we're starting with number 7 now. The female part of the plant is called the carpel, which is made up of the stigma, the style, and the ovary. Look for a central stalk. You have identified the style. Remember I said you should have this part left. Mine came apart, but it should look very similar to that. All you do is the same thing. Grasp it at the base. I've got mine kind of at the middle. Grasp it at the base and just pull straight out, and it should come off on its own. That is your stigma and your style. Okay, look at the tip of the style, which has a sticky end called the stigma. So if we're looking here, this would be the stigma. And if you put your forceps on it, or even your finger, you can kind of feel sometimes it has a sticky, maybe a sticky fluid to it. Okay, why do you think the stigma is sticky? Well, what's the purpose of the stigma? The pollen stamen are like this. The pollen would fall off into this section. This is a hollow tube, so the pollen would travel down the hollow tube into the ovary, which is this part down here. That's where the ovules are, and it would fertilize the ovules. So why do you think the stigma is sticky? Well, we need to keep the pollen on the style. It the stigma and fertilize the ovules. So remove the stigma and the style from the flower and locate the ovary. You're going to gently pull it open with your thumbnail and forceps to observe the ovules. Okay, so watch what I do first, and then you can try to do it. I'm going to take my thumbnail, and I'm going to push it in here, just kind of using it like a knife to cut it. Okay, and I'm going to gently, ever so gently, open it up. And when I do that and you look inside, you should be able to see little nodules in there. Those nodules are the ovules. Okay, so this swollen part all the way around is the ovary on the outside, and inside you will see the ovules, kind of on a little stalk, it looks like. So let's draw that picture. It says draw your findings and label your drawing. So we're going to talk about the ovary, and inside the ovary you have this little stalk, and on that stalk you will have the ovules. And my stalk and my ovules are a little bit lighter green than the ovary is, so I'm going to color those just a little lighter green than the ovary itself. Obviously, you can take your time and do a better job. I'm trying to do this quickly. And then the ovary is going to be a dark color. Again, take your time, color it nicely. So this is the ovary, and this is the ovule. Okay? All right. Very last page that you have is the critical thinking questions. And so when we talk about the flower, the question says, when people visit an apple orchard, why do you think the farmer asks visitors not to pick the apple blossoms off the tree? Well, what do the apple blossoms eventually become? They become the fruit, right? So if you pick the apple blossoms off the tree, that means you're not going to get as much fruit from that tree. Okay. Number 2, why do flowers that are pollinated by insects have large, brightly colored petals? Well, insects like flowers, and the brighter the color, the more they're attracted to it. And so this is a way of luring in insects. or even other animals. Okay, and then number 3, why do you think a flower has more pollen grains than ovules? Okay, so pollen, of course, is carried by wind or insects or water, and a lot of times does not necessarily make it to the female parts of the flower. So the flower has to make more pollen than it does ovules to increase its chances of fertilization. Okay, and then there was a 4th question that I added to this that said describe how pollination happens. Be specific. So when I say be specific, I'm wanting you to give me the parts of the flower. So we say that pollen lands on the stigma. It travels down the style. Well, even before we get to the stigma, where does the pollen come from? Pollen is produced by the anther, right? Then it lands on the stigma, then it travels down the style to the ovules and the ovary. The pollen fertilizes the ovules, which become seeds. And seeds are the end goal, right? But before we can do anything with the seeds, the ovary is going to swell and become a fruit, or a ripe ovary, and then it disperses the seeds by either rupturing or by being a fruit that another organism will eat, and then the seeds go through the digestive system and are deposited somewhere other than that area. Okay? All right, you have now completed the flower dissection. Everything that you have pulled off of the flower can go into the trash can. Grab a Clorox wipe and wipe down the pad and your instruments and put those back where you got them from. I hope you enjoy this dissection. Like I said, it's our first of many, and I will talk to you and see you for our next dissection.